I'll take that again. Good morning, Ghana. Good afternoon, Nigeria. Good afternoon, East Africa. Good evening, Asia. Good day from any part of the world you're joining us from today. My name is Musa Achibugu Dixon. I'm a professor of pharmacological biochemistry and the director of Trans-Saharan Disease Research Center of IBB University, Lapai, Nigeria. I'm also your moderator for today's webinar. You're especially welcome to this edition of the TDRC webinar series. Ibrahim Badamasi Babangida University, Lapai, Nigeria, is a relatively small university, but it is doing great things. I'm trying to put Nigeria on the world research map. For instance, we're the first to carry out a seroprevalence study on COVID-19 anywhere in this part of the world. We did that research in April 2020, and the result was so interesting that the body responsible for such research in Nigeria adopted our protocol as a protocol for carrying out seroprevalence study in Nigeria. It is based on our research strength that the university management established the Trans-Saharan Disease Research Center in collaboration with researchers from Francis Crick Institute in London, with a vision to be a world-class center of excellence for quality and innovative research and capacity building in Trans-Saharan disease. The center has a mission to promote the awareness and eradication of Trans-Saharan disease through science, technology, and partnership. It is in furtherance of the goals of the center that we have started this international webinar series to curate uniquely African ideas that can be used to provide solutions to the African health challenges and reduce the African disease burden. This is the first of the webinar series, and we hope to make it a regular event. Today, we are privileged to have the chairman for today's webinar, a pharmacist by training, and an accomplished researcher by the name of Professor Martins Emeje. He is the head of research and development in Nigeria's foremost pharmaceutical research agency, the National Institute for Pharmaceutical Research and Development, NIPRID, in Abuja, Nigeria. He's also the founder of the Nanomedicine Laboratory in the same institute. So now I'm going to hand over the mic to the chairman of today's webinar series, Professor Emeje, to take it off from here. Chairman, sir.
Okay, thank you, Professor Musa. It's nice to be here, and I welcome everyone to this seminar. This is an international interaction. I'm not going to spend much time because there is going to be, we are here to listen to our guest, Ferez, who is an accomplished person in his own right. He has over 400 citations. He is a principal investigator. He is a founder of Life Sciences Institute of New Jersey. He's into a lot of research and development activities, and you may just find time to look at who he is on the World Wide Web. He has over 13,000 followers on LinkedIn. LinkedIn is a professional social media link that connects people that want to get things done across the world. In this part of the world, we are people that are challenged by our environment. We are people that think outside the box. I want to commend Dixon and the university, IBB, for this for thinking out of the box, for this exercise, really, for what they have begun, because this is what Africa needs to do. And I'm happy that we have someone in the status of Ferez coming to speak in this first series. It is my candid opinion and a prediction that something great is going to happen as we begin this series with this university. The size of a university is not relevant, neither is the size of man relevant. It is what you make, it's what you produce that makes you relevant. And I believe that that is what IBB University is showing the world and showing the country. It's not just about Nigeria, it's not just about Africa, but of the world. It is, you know, you have to be courageous. You have to have the competence and the capacity to be able to do what Dixon and his university have decided to do. Because if you are timid, if you don't believe in yourself, if you don't have confidence, this is an area that people are running away from because they believe that it has to come from where Ferez is coming from in order for us to think that it's of value. And that is what Dixon and his group and his team and his university, I believe, are not ready to take. If we cannot continue to be in that way, and we'll be listening to Ferez as he exposes that. Africa, for me, is a home of commerce. What do I mean by commerce? When people talk about Africa being full of infectious diseases, I say yes. And that is why we are also the home to it. For example, you cannot claim that you have a drug for any disease that is infectious or of origin without coming to us for clinical trials. You know, so if we refuse to allow you to do clinical trials, then your drug becomes useless. So that I have the disease means that I am important, you know, but we also know that for every disease, the solution to the disease is in the environment. So the people that live in that environment, especially those of us who are scientists, if you do not use your knowledge to get this across to your people, then you are in trouble. And that is what I believe this series of webinars is about to kickstart. So it's my candid honor and privilege to chair this session, but I would like to yield the floor to Professor Musa, who is going to do a brief bio of Ferez, and then he takes over for his lectures for about 40 minutes. Over to you again, Professor Musa.
Thank you very much, Mr. Chairman, sir. Yes, our invited speaker for today is Dr. Ferez Soli Nallaseth. Dr. Ferez Nallaseth is an experimental and conceptual biologist who either has worked or is working in a broad spectrum of 19 fields of the life sciences over the past 48 years. Yes, you heard that right, 48 years. His work ranges in complexity from degradative NaH plasmids in soil bacteria to adaptive neurophysiological responses in turtle brains. However, it was the core of Dr. Nallaseth's work on the principles of evolutionary genetics or speciation, specifically species incompatibility, resulting in the dysregulation of murine chromosomes and genome biology that was reflected in traits such as aberrant XY sex determination in mice, which is continuing to make the most significant contribution. This background and its integration of synthesis of multiple fields has, sorry, let me admit some people to join us, has led to Dr. Nallaseth recognizing the single greatest but surmountable challenge in medicine and biomedical research, namely supplementing the current necessary paradigm of responding to immediate pathological conditions with its disadvantage of perpetuating genetic disease conditions and expanding patient populations. This is largely a consequence of exclusively managing these pathological conditions of genetic disease lesions, rather than preempting their insertions into the genome. This latter approach is made possible by initiating the integration and synthesis of vast body of literature and methods, including an array of subfields in the life, physical, and computational sciences, as well as in mathematics that have attained various stages of development over the past 150 years. This approach has been recognized in the published manuscript as a combinatorial approach in real-time operation, CARTO. This was published by Dr. Nallaseth and his colleagues. To promote these objectives, Dr. Nallaseth has founded the Life Sciences Institute of New Jersey in 2004. However, Ferez recognized that this undertaking requires a collective and collaborative effort based on comprehensive diversity. In this effort, he was joined by board members who represented a globally diverse spectrum of location as well as an array of distinct fields. Thus, in November 2018, Dr. Nallaseth, as its founding president, and his colleagues co-founded the current restructured LSINJ. Dr. Nallaseth has done his work in or through collaborations in universities and research institutes across the United States from 1976 onwards, including with or in top-tiered institutes such as Caltech University, MIT, Roche Institute of Molecular Biology, Fred Hutchinson's Cancer Research Center, and Mount Sinai School of Medicine. He attained the rank of research professor at the Florida Atlantic University in Boca Raton, where he collaborated with the neurophysiologist and chair, Professor Peter L. Luz, as well as the director of the Center for Cell and Molecular Biology and Biotechnology, Professor Herb Weisbach, who is a member of the National Academy of Sciences, USA. In doing so, Dr. Nallaseth has successfully operated technically complex biological systems, qualified, corrected, or redeemed work dismissed as non-reproducible in multiple fields, and published in top-tier journals such as Cell, Science, Nature, PNAS, MCB, and Journal of Biochemistry. Perhaps Ferez's most recognized publication came in 1981 while working as a technician in the laboratory of Dr. John Wilson at the Medical College in Georgia, in Augusta. Ferez successfully executed the first direct test for prenatal detection of sickle cell anemia. This enabled Dr. Wilson's laboratory to outcompete frontline laboratories at Harvard and the University of California in San Francisco. These results were published in PNAS. In 1987, Ferez was awarded his doctorate PhD degree in biology from the University of South Carolina in Columbia. In 1979, was awarded his master's in microbiology, and 1977, bachelor's in microbiology from the University of Georgia in Athens. In 1975, he earned a bachelor degree, BSc Chemistry from Bombay University in India. Dr. Nallaseth and his colleagues in the LSINJ hope to make a significant, necessary, neglected, and maximally impactful contribution in medicine and beyond to humankind. This is by going beyond advocacy and into the operational integration of CARTO in preempting genetic disease lesion and the perpetuation of recurrence of pathological conditions that they cause. Ladies and gentlemen, join me to welcome Dr. Ferez Suli Nallaseth to deliver his lecture. Thank you, Ferez. Ferez, unmute yourself, please.
Yeah, I just did. Thank you very much for your generous introduction, both Professor Amajay and Professor Musa. It's a great pleasure to be over here with you in the morning. I might have a slightly raspy voice. Please pardon me for that. I've taken lozenges. I look forward to this morning with great enthusiasm. I haven't been to a meeting in a long time, but particularly since Professor Musa, who is an integral part of our institute in moving it around, moving it forward, has been so generous in inviting me over here. Professor Amajay has been so kind with his words. I hope I live up to his description. I have connections with Nigeria that go into a passion of mine from many years. I see that you actually got my squash shirt on that picture. It's a little old, as you can see. The picture is a little old.
You are not sharing yet, Ferez. Please share your screen.
Oh, let's see. Can you see this?
Yeah, it's coming up.
Should I go full? Let me go.
Yes, you come down, down, down. Not there. Come below.
Okay.
Yeah, yes, yes. No, no, no.
Oh,
no.
yeah.
Yeah.
Yeah, okay, great. Can you see
Good.
this now?
Yes, we can.
Okay, great. Did you hear me earlier?
Yes, we did.
Okay, thank you. So, so basically, we are very pleased to be here. I was talking about my passion. I know Nigerians from way back. One of my closest friends and mentors was Francis O'Day. He was the #1 Nigerian player and an internationally ranked player. We had many beers and talked a lot. I went to his club regularly. So I go there. I know about Nigeria from before Francis, and I'm very, very glad at least to be here on a virtual basis. So I want to start by explaining the approach that I'm taking today. So I need a clock somewhere so I don't go over. You know what? Okay, so I have 6:24. I mean, I guess it's 12:24 your time. Dixon?
Yeah.
It's about 12.
It's 12:24 here. We are 6 hours ahead of you.
Yes. So I'm trying to see how much time I have. I want to make sure that at least I touch the top end of these of the slides I have. So I have many slides over here, and the reason is very simple. To put this,
you have 40 minutes.
Okay, thank you, sir. So so the reason is very simple, and it also explains the resistance that this work or this approach has had. There's about 150 years of work. If you do Google on any one of the fields that you put together, there are hundreds of millions of papers, data pieces, methods, etc. I don't know them all, but I know they exist. And if you apply a standard of measure or rigor that is applied to a narrow field, let's say, or a narrow area, let's say the Sputnik of molecular biology, which is the activator repressor complex at the lac repressor in E. coli, you'll never understand the others. You never rediscover America. You never discover penicillin. You never launch Sputnik. So the basis is that if you put all of these together, someday, somehow, some things will emerge. Now, you don't get funded for that, which is why all of us contributed to inaugurating the LSINJ. And the title of my talk, I guess you've seen, and Dixon is a big part of it. For convenience, I got the wrong I apologize. This is the wrong PowerPoint. That's why it's not. Uh, please pardon me. I'm going to go back. I made edits last night.
If you want to change the PowerPoint, you may have to stop sharing this, then you share the other one. Except
Okay.
Yeah.
Let me just, I was trying to change a slide before we talked, and I suddenly realized that I'd made many changes. Okay. Actually,
So while Ferez is changing the slides, as soon as you change your slide, just share and we'll give you the podium again. But while he's changing the slide, the African scientists that are on this platform, I'm sure that one of the questions you have been asking yourself is, okay, he's back.
yeah, I'm having
Okay.
trouble getting it to full screen.
Yeah, just repeat
down,
what
down,
you did
down.
earlier.
So this should go to full screen. Okay, share, new share. This is the one.
You come down to your right, below the screen. Yeah, come to the.
So I'm here and I'm on share.
Yes, we can see, but it's not maximized. Come down a little.
Okay. So I don't know why this is stuck.
Okay, press F5 maybe.
Right, thank you. Yeah, okay. I don't know why it's not changing. We'll just keep going at this stage. It doesn't matter, it's a minor change. I turned all this around and okay, let's just keep going, sir. So for convenience, the whole overview is that there are networks of genes that maintain and repair genomes, whether you're COVID, whether you're SARS, COVID-2, or a human being. You make the genome makes errors, the environment introduces errors, and there's an insertion of genetic lesions, and you get a disease pathology, right? Just to give you an idea, this is a wrong number. It should be 2.8 times 10 to the 18th. I corrected that. Mutations in an average lifetime. So what I will do is I'll send the correct numbers to Dixon and as a PowerPoint, and it'll be an improved version of this. This is the beta globin gene. I'm going to try to rip through it very quickly. All this is published and old work, but it lays the basis for what we're talking about. It causes sickle cell anemia, and 92% of the cases of sickle cell anemia are in Africa. So the beta globin gene encodes is beta globin. I'm sorry, the HPB locus consists of the beta globin gene and the alpha globin gene. There's a locus control region that controls the expression and switching of all these genes. They form a dipeptide with the beta and the alpha locus. When there's a mutation and the A at position C changes to a T, it goes from glutamate to valine. So as biochemists, you all know what that does to the properties of the gene. I'm going to leave alone all this. It's there for you to see it. That mutation causes the protein to not bind oxygen with a high Kb. The oxygen is relieved. The HbS fibril aggregates, and the cells are fragile. They lyse. Okay. A single mutation in the HBV locus is protective. It resists malaria by 90%, and early survival increases 60% over the homozygous case. So you can see the power of that mutation. And this is so if I could move this down a little so you could see this. So this is gene therapy that was used to correct the HBB sickle cell mutation. The wild type gene was, along with the locus control region, was introduced into a disabled vector in which the GAG pole genes were mutated, so it could never infect the patient with AIDS. The patient's stem cells with this construct were amplified in culture, 85% expressed. It was put back into the patient who was treated by chemotherapy. The patient was Ms. Janelle Stephenson. And even after 9 months of gene therapy, there was no sickle cell. So this is before gene therapy. You can see the sickling effect. And this is after gene therapy. So they're nice biconvex discs, right? So this seems like a great therapy, and it is for the immediate person, right? But let me go back. So gene therapy came after 100 years of work on the hemoglobin locus, right? And so you have to imagine that this highly technically accessible system took 100 years, and the actual gene therapy protocol took about 40 years of development. It's a fluid system, it's a blood system. It's not like the brain or the heart, which is highly organized. There are about 5 million red blood cells that are produced and turned over every second. Okay, there were other problems with this, but the main problem is that after you done gene therapy, first of all, you'll never get to the extant population of 20 million globally. Most of them are passing, and there are reports that the older population is not being looked after. But after you got to them, there'll be a new cases of that A to T transversion or missense mutation causing sickle cell anemia. So do you want to discontinue therapies like this, with CRISPR-Cas9 and other methods? Of course not. They're transitional methods. They're necessary for here and now, but you have to plan for the long term. So the CARTO is a slower long-term method, but it stops these mutations eventually. To do that is work that will need more than one country and one generation, right? The LSINJ proposes this. So, I'm going to come back to this later. This is actually my experiment, and I can tell you, we did in collaboration, but I got it to work the first time. If this experiment had not worked, I would not be a scientist today. And why it worked remains a total mystery for me because of the odds technically and my maturity as a scientist. At the time, I was a technician. So as I don't know now what to anticipate in the next slides, I'm going to tell you this result right off the bat. This is the wild type sickle cell. So you get 2 fragments cut with, so this is a southern blot. I guess I have to tell you what a southern blot is. Basically, if you want to analyze any gene, you cut it with genomic DNA, cut the genomic DNA with restriction enzymes, separate the fragments on a gel, transfer it to a blot, either by electrical methods or by capillary action, and analyze them with a probe, which is the part of the gene that's been cloned, wash it off, and so you get a single band, right? So that's what these bands are. The restriction enzyme over here was DDE1. It cuts at the sickle cell site. So the sickle cell mutation eliminates this this A. Then it will not cut and you get a 376 base pair band. So this is the heterozygous trait, and this is a homozygous disease. You can see that it's not a pretty gel, but it did the job. I'm sorry, this is the homozygous disease of 376 base pairs. Let me try and move this. So this is a 376 base pair band, right? So why do we need this approach? There are a minimum of 13,700 Mendelian genes that cause diseases. About 65% of all diseases are caused by a genetic component. We are excluding, this is a minimum, multifactorial polygenic high complexity diseases. The costs are something like $47 trillion in 2 decades, as recognized by the World Economic Forum and by a Harvard Public School study. That's a conservative cost, right? There's a prohibitive cost of ignoring issues addressed by this combinatorial approach to preempting the mutome. The mutome is any complex, any collection of proteins and nucleotides or any environmental factor that causes a mutation. So it's a term we just coined. Okay, so this will come in the end, but I'm basically saying that all levels of organization of the genome from a single nucleotide to Mars, SARS are under genetic control. Okay. So I'm going to keep moving to the next slide. There's a fundamental premise in this. If you have an interspecific hybrid and it's introgress, so any species, this is a foundation of evolutionary genetics of speciation. If you have species A and a species B, they can mate productively to give you an AB genome. There are all kinds of errors in that genome, so it's a source of raw materials for analysis with the power of modern methods. CRISPR-Cas9, GWAS, all the kinds of things that you talked about. You can look for these methods by panning. In other words, when there's a mutation in one gene, other genes are also affected, and that's known from a large body of work. The combinatorial approach has a discovery and an analytical phase. Okay. I'm going to skip over this. There are many things that we need to go over. So this thing is completely changed. Okay. So the first thing that I'm going to be talking about is sequence instability and functional inactivation of ferro Y chromosomes can occur in a specific genetic inbred strain. Okay. This is a mouse that was detected by my colleague Barry Whitney at Jackson Labs, and then Dr. Eva Eicher also did some work on it, right? She characterized it further. I showed that it was genetically unstable. This is the paper that we published. I'm sorry, this is the Southern blot. So this is a paper that we published in 1992. It was very controversial because it was thought that the inbred strain is completely stable. And I'm going to skip many slides there for you and send the PowerPoint to Dixon later. So just to give you this is one of the fragments I cloned. This is some background over here. This is an underexposed version of male DNA. So it's cut with EcoR1 and probed with this clone version of this fragment. These are all X-linked fragments. This is an alpha globin fragment, and these are these two fragments, 2.2 and 1.7. So this is an XO mouse, and this is an XX mouse. It's X-linked, okay? We quantified the ratio of this band to this band or this band and came up with the red and green bars, and you can see, and this is really an underestimate because we don't have these exposures right now. You have to expose these blots so the pixels can be resolved by the densitometer. But at this level, there's absolutely nothing. It actually goes into negative numbers, okay? If you take that YPAS mouse, that Swiss mouse, in the same background, let me get backtrack a little. That Swiss mouse in the 129 background, over here, here, and here are the X-linked fragments, and this is the Y fragment. Mate this with another black 6 mouse. So 129 mouse is a standard laboratory strain. You made it with the black 6 mouse, and now these are the two standards. And over here, 6 of 8 mice have a 600 base pair deletion. And these are female mice. We can't use male mice because this band will totally cover up everything, right? So it's an assay, and now you can see all the bands over here and the yellow bands coming up. So this is for this plot, and this is for this plot, and you can see the yellow bands all coming up in intensity. This is another kind of fragment. This is a translocation. This translocation is 2.2 and 1.7. This is an XY female, and these are different tissues from that female. So I'm pointing to the bands and not telling you the whole process because it will take too long. And what I needed to do was to show you the whole basis, molecular basis of this is completely wrong. Dixon, I'm going to have to stop sharing this screen because a lot of the slides are not there. I don't know why it's not showing up.
I can imagine you've not had much sleep. Sorry, please.
No, it's so this is the one that I wanted, that I had it on last night. And I don't understand why it's not going on. I apologize for this fumbling. Do you see this? It's the same thing that's coming up. Okay, I made a mistake. So let's just keep going. I apologize for this. Okay. So basically,
Can you press F5, please?
Okay, sorry about that. You get it? Okay, let's just keep going. There's a fundamental. Okay, so we showed epigenetic changes in could make a difference. Basically, we did PCR. I guess everybody is familiar with PCR. This is a fragment. These are primers that can stick to it, and the enzyme will copy it multiple times and amplify it, right? So the reason why we did PCR was that we also had evidence that there are some epigenetic changes that were ongoing in this chromosome that came from a wild mouse. So we designed this test. Basically, I did this with Peter Hornsby at the Baylor College of Medicine. This is the clone probe in genomic DNA. I'm sorry, clone probe mixed in with genomic DNA, which is female. So it doesn't have the sequence. This is a gradient of buffers of increasing pH at 56 degrees. And this is amplified with DCTP. This is amplified with 5-methyl DCTP. And now you can see at 10.65, there's a shift, and there's a total shift at 11.15 pH at 56 degrees. It's going up gradually. With methylation, everything stays rock stable and it shifts asymptotically. We applied that to some females that we had shown, XY females had undergone a shift, and this was very controversial at the time that we published it. But these are two XY sisters that came from the same litter. So you cannot have radical changes in the genome, although you do get somatic mutations. These copies are 100 copies. All 100 copies have essentially shifted and melted and gone up to 11.15 in this sister, which is XY. And in this sister, you're starting to see super shifts. Melt this blot and probe with 2 other Y probes, and you see oscillation and shifts in this female, but it stays fairly stable up to 10.55, then shifts up and melts completely at only at 11.15. This female, you have the super shifts. It starts shifting at a much higher pH. It goes down completely, and you see all the melted DNA up here in the screen. So feral Y chromosomes, there's other data that we have that I'm not showing. Backcross into inbred strain genomes undergo epigenetic modifications that lower double-stranded stability. In addition to the genetic modifications, this has implications of YSRY, which is the gene that determines sex expression, sex determination and gametogenesis, XY heteroduplex formation, sister chromatid exchanges, which on resolution uncouple Y linkage, induce deletions in the SXR sex reverse region, SRI sex reversal sex region, sex determining region Y chromosome, and other loci. These results may explain lowered introgression of the Y dom chromosome, the mus domesticus musculus, as compared to the Y mus Y chromosome, the mus domesticus chromosome, in natural hybrid zone in Eastern Europe and Western Europe, right? This is a probe and work of Robin Lovell Badge. He generously gave us this locus, this 3.5 kb fragment. I subcloned all the fragments and did some work on this to show that this is also unstable, okay? So I'm gonna zip through all this. Basically, if you have an inverted repeat locus, like the SRY locus, you can get these structures, which are holiday junctions, and they're highly unstable, okay? Basically, this is an inbred strain mouse, the same black 6 mouse inbred strain that we are talking about. It's cut with, we're looking at 3' side, cut with bagel 2, HEPA1 over here, a bagel 2, MSP1 over here. These are the predicted fragments. This is liver DNA, and you get the right size fragments. These are in all circumstances deletions, because the smaller size and the fragment is being cut. So the restriction size is being retained. Over here, it's female DNA, so it's not hybridizing with them. This is sperm DNA, and you can see a faint band going down here. And if you go to the testis from this male mouse, you see these bands, but you barely see anything over here. You can see it on the other end. In addition, there's another deletion, and this will feature prominently. This deletion is present in all combinations of mice. The 5' side is not showing up. You cut with PST1, HEPA1, or Hindi 3 HEPA1, and you see a deleted fragment over here, okay? There is a modulation, genetic modulation. In other words, the background determines the frequency of these deletions, okay? This deletion that I showed you over, these are the predicted fragments earlier. We sequenced cut out of a gel sequence in Leanne Shine's lab at Rutgers University. It's amplified. The amplification center, by cutting the different enzymes, is on the 3' side of the gene. The gene is this HMG box. If you cut with different restriction enzymes and hybridize with the probe, you'll find that most of the hybridization is on this side. There is a inverted repeat and possibly some replication signals over here, but we don't know if they work. Right? There's also a preference of a genetic background on which this amplification occurs. Okay, so I'm going to leave all this alone. We don't have time. This is the data that gave us those results. We also did PCR with genomic DNA, and this is the locus. Okay, I'm trying to shift this. Okay, so this is the locus, the sex determining locus. This is HMG box. These are Robin level Badger's primers. They're called breakpoint primers. In one deletion that they thought they were making, this 11 kb fragment is deleted. It's present in wild type. So if you amplify from these primers, you will get this fragment in the wild type, but not in the mutant, XY female mutant. So this is the negative control. We made the amplifications with long template PCR, ran them on a gel. This is female DNA over here. These are markers over here. This is male DNA. And you can see the 11 kb fragment in the males of normal size. We don't know what these are, whether they are generated in the PCR, but we know from southern blots, these exist. So you can see fragments up to 2 kb, but you can't see anything at less than 2 kb with this probe. On the other hand, if you use these primers as probe in the same mice, low exposure over here, so you can see all these bands, and high exposure over here, you can see amplification up to 11 kb over here and here. But you also see fragments that are concentrated in the male DNA. This is an autosomal amplification. It's lower in female than male. Why is this happening? Because this primer binds all over the genome, in addition to the SRY locus. The enzyme corrects the mismatches and produces all the other fragments, in addition to this fragment, right? So if you do it at a higher exposure, if you quantify this fragments densitometrically and look at the ratio of the 0.95 to these fragments over here, you get something like a 450-fold amplification. In this mouse, and in this mouse, you get about 200-fold, whereas other fragments are non-specific. So I'm going to leave this alone and basically move on to say that this locus is highly unstable, exactly as our work on the SXR region that surrounds it showed. Okay. I'm going to drop all this. This is just a fusion fragment that we pulled out from the Lambda library, and it's not necessary. So this is the SRY locus is an inverted repeat, or it forms these structures that are unstable. We had already shown that the SXR region that spans it is highly unstable. So what happens? This work taken from Ian Hickson and Susan Glass's laboratories. When you have a replication fork, this is a leading strand, this is a lagging strand. Everybody knows that DNA is synthesized in the 5' to 3' direction. These are extremely well characterized proteins. This is a model. When there's a break in the strand shown by the star, if the break is repaired, then replication will continue normally, and you get a recovery and resumption of DNA replication through a classical pathway. If this break is not repaired, this strand is extended like a chicken feet, or the classical holiday junction that I'm about to show you about. That occurs at this, that would occur at the SRY locus. So here's an assay that I developed in addition to EMSA assays. And I did this work in Steve Brill's lab at Rutgers. So the assay is very simple. It's a streptavidin, biotin, pull down assay. This is a holiday junction. It's made of about 20 to 30 nucleotide fragments that are annealed in a lab. Steve designed these fragments. In this case, there's no biotin. In this case, there's a biotin, the red bubble. Okay, this is streptavidin beads. Sorry, this is a protein that binds to the holiday junction. But this protein, this complex, will not bind to the streptavidin beads. This protein will bind to the streptavidin beads because it has biotin. When you wash it, this will be the signal in the control, and this will be the signal in the wild type. So these are an array of structures that form a replication and recombination intermediates. This is 1/5 of the protein. This is an RMI protein that is part of a triprotein complex, and now 4 proteins. It's called RMI STS helicase top 3 topoisomerase. It's the analog of the human Bloom syndrome complex that causes cancer. Okay. This is the background. C stands for holiday junction. This is without DNA. This is with the DNA that does not have biotin. This is with the holiday junction DNA that has biotin. This is a similar DNA called Nick holiday junction. And the condition in which you wash this, bind and wash this, is very important for this result. It has to be 150 millimolar bind and 400 millimolar wash. These are all the other structures which are background. We normalize this to this and put the blot over here so you can see it. And this lane is a control, which is a quantitative control. It's not washed. And you can see the signal over here of low exposure and high exposure, and this is the mean. Okay, normalize. Everything is normalized to this signal. This is the control, negative control, no binding. This is a holiday junction binding over here. This is a holiday junction, low exposure, high exposure, mean. And this is a next holiday junction. It's at least 6 times higher than the next holiday junction. So this is a highly, I don't want to spend more time on this, but show you what the results are with the 3 protein complex. That was the first protein. These are the controls that work well. This is SGS1, the 322 base pair N terminus bound to the holiday junction and then competed with RMI1. They have different tags, right? So RMI1, 5, 25, 100. So you can see about equivalent binding at 5 and 25. There's a little depreciation over here. This is top 3, 5, 25, 100. After RMI1, 5, 25, 100. And we quantified it over here. And this is RMI1 and top 3 on SGS1, 5, 25, 100. And you can see the quantification. All the others go down. All the others go down. This one goes up by 200-fold. If you use the shorter fragment, well, just I'm sort of if you use a shorter fragment, you get the same result. Okay, the shorter fragment is over here. Let's see. Okay, the shorter fragment is over here. Can everybody see this? Dixon, can you see this? Hello?
Yes, we can see. Go ahead. Please.
Yes, we can.
Okay. Okay, so this is another assay. This is competition. This is called EMSA. Basically, if you label a probe and it binds to DNA, I mean protein, the mass to charge ratio change, it shifts up, right? If you have a non-specific competitor, it will not shift down. If you have a specific competitor, sorry, so it shifts up. If you have a specific competitor, it disappears, okay? And this is the probe that is bound to the protein and super shifted with antibody over here. Okay, so that's the electrophoretic mobility shift assay. This is the probe with Holiday junction, competed with Holiday junction at about 100-fold femtomoles. The probe is 50 femtomoles, starting to break up, has gone by 300 femtomoles. If you look at single-stranded DNA, it's already right from the beginning, it's starting to break up the complex. It's all done in the same test tube. The complexes are made in the same test tube, and the competitors were added in separate test tubes. That's right. So it's highly stable, and you can see it over here. If you do the reverse experiment and bind the probe without competitors, you can barely see a signal at maybe 1,000-fold nanograms of RMI, whereas with Holiday junction, you can see it easily forming at 125 and completely formed at 250. So it's Holiday junction that is recognized by the RMI and also by the complex by the pull down assay that I showed you. So we have this model. Basically, if you have these three proteins shown by the colors, top three RMI and SGS1, they'll bind to the Holiday junction. We propose that the RMI1 binds first and the others dock together, okay? But it needs both RMI and top three to pull the SGS1 on it. The structure itself promotes the binding, okay? So it's not just the protein, it's which DNA structure and which protein binds first. So this paper is now published. It's another assay that we have, and I think I'm going to drop this. It's a replication assay with viruses. I did this in Mel DePamphris' lab. It showed that when you combine the BPV genome with the polyoma genome, it suppresses replication, but not, I'm going to drop all this, come to the combinatorial approach, because we only have about 5-10 minutes, right? So the BPV genome, bovine papillomavirus, did not regulate the polyoma virus, but did suppress it. And we did a series of mutations. Okay, so what is the combinatorial approach? I already mentioned that hybrids create problems for many different genes. There's a broad spectrum of lesions, okay? These lesions, mechanisms reproduce context of insertion of genetic disease lesions into the human genome. For example, failure or uncoupling of co-evolved gene, regulating the maintenance or repair of genes for the substrate genes, chromosomes, and genomes. For example, when the HIV gene or the COVID-2 gene, COVID-2 race gene, or ACE gene mutates, it's not that the COVID-2 or HIV is mutating it, it's the cells genes and proteins that are mutating it. So controlling those gives you a process that someday can stop those mutations, the variants they generate. How do you look for these genes? It's called non-selective panning. I mentioned that already. It's like a combination of GWAS and saturation mutagenesis or secondary structures of protein that you screen for, and the Large Hadron Collider. You know, these are all unknowns. These all look for unknowns with different processes and come up with knowns. Okay, so I'm going to leave this alone. It is this combinatorial approach in real-time organization is the only way to identify networks of co-evolved genes, maintaining and repairing genomes which fail and uncouple from the substrate genes. Give you the insertion of genetic lesions and disease pathology. How do you do it? I think the paper that we listed is a better way of looking at this. It's over here. Over here, you should have combinatorial approach. You have 4 different segments that are overlapping. They don't have to be done in series. At first, you have evolutionary and population genetics of speciation, mouse genetics. Then you look for low density screening with omics or with coat colors or viability of mice. You go to higher resolution screening with optics, multispectral optoacoustic tomography. You use supercomputers. And then when you have this working in mice, maybe 100 years from now, you can start thinking about going to humans. You can look for errors in intracellular processes that are going on in a real-time basis with a lot of these methods. You can look for transmission of these signals from the cells by various methods like radio waves, wireless, fiber optics, cell phones. There's a mutome failure rate that will pass. So then you start looking through, and this is all automated in computers with data analysis, supercomputers. There's an analytical phase, activation of intracellular regulation, which has been identified in mice with these hybrid mice. The mutome is repaired and you come back. So this is real-time operations, right? I think I've told you this right at the beginning, the enormous costs of not doing this work. And these are all the quotations from Einstein and others, why this is not done. There are serious challenges. I'm not naive. There's serious conceptual, scientific, technical, ethical, and economic challenges to that. But the real barrier is not those challenges. The real barrier is what Einstein and Feynman, Barbara McClintock, Robert Oppenheimer, and his Soviet counterpart, Andrei Sakharov, have identified. In other words, there's a scientific culture that stops this. Okay? So I'm going to stop this. These are all our previous slides. I apologize for this, but maybe there's an accident that cut me short, and I'm happy to take any questions that you might have at this stage.
Okay, thank you very much. In this part of the world where we are, we call it Africa. The headquarters of where this webinar is streaming to the world this afternoon is Nigeria, and Nigeria is the heart of Africa. Nigeria is the largest economy in Africa. Nigeria is the largest Black nation in the world. Nigeria is the largest Black democracy in the world. And Nigeria is also proud to be the eye and window through which the world sees Africa. These are undisputable facts. We are happy to have listened to Ferez this afternoon. I believe that two things happened this afternoon. It's either you are one of us, and so you were able to follow Ferez very well, or you are not one of us, and you actually did not understand what he said. You not to worry. For the next 60 to 120 seconds, I will convert what Ferez had said into the language that those of you who did not understand him will understand.
Mr. Chairman, just a moment, sir.
Okay.
Ferez, please, can you stop sharing your screen so that we can see the speaker?
You know,
you want to see me?
Yes. Ferez, can you stop sharing your screen, please?
Stop sharing?
Yes.
Okay.
Thank you.
Oh, okay. Now I can thank you.
Okay, so can you see me now?
Yes,
Yes,
sir.
sir. Yes, sir.
Okay.
Yes,
Yes,
Prof,
I saw somebody who sent a message that we cannot see the speaker. Don't worry, you can hear the voice of the speaker. I'm one of you. But it's okay.
Prof, we can't hear you. You're muted, though.
Who went to mute me now?
No, what happened was you were muted. And
okay,
I
can
tried
you
to
hear me
unmute
now?
you. Now I can hear you.
Okay, thank you.
Sorry, yeah.
Network of genes responsible for maintenance and repair of genomes. When you hear of genes, for those who don't know what genes are, if you are tall or you are short, if you are black or you are yellow, if you are green, if your hair is white, if your eye is blue or is brown, if you talk too much and you don't talk too much, if you drink too much and you don't drink too much, if you like many things and others don't like many things, all of those things that define people's, either your character as they are in you or that you express them, are all carried by what we call genes. So if you are on this webinar this afternoon, even if you cannot remember anything, remember that this webinar talked a lot about genes, the role of genes in how man could become a better species, that is, in essence, how to treat, how to take care of diseases. And when we say treatment of disease, we mean, how do we identify disease? How do we locate disease? How do we get preparations that will take care of the disease? Okay, that's what we summarize by disease. So this is what we call genomic approach to drug discovery and development. Because the fundamentals of disease treatment is diagnosis. So science currently tries to look at the genomic approach to disease diagnosis. And a lot of what Ferez has talked about this morning had to do with genomics and how you can use genomics in diagnosis and prediction of treatment. Now, this is basic science as foundation for development. In drug development, we say drug discovery and development. And the development, we say translation. Translation is when we utilize the knowledge from drug discovery in order to get products and services out there which our people can use. What our people are interested are the services and the products. What we heavily work on is the basics of it, the science of it, which Ferez has dwelt on this afternoon. He talked about mutation in lifetime, and he noted the millions of mutations that have occurred in lifetime. He looked specifically at sickle cell, and that was interesting because if for those of us who don't know, you only know that somebody is a sickler. And for many of us too, I want to use this opportunity to tell many of us too who think that if someone is a sickler, you are not supposed to get close to the person. It is not a communicable disease. Sickle cell, and from what Ferez presented this afternoon, let me bring it back further home. You saw something on his slide, one of his slides, where he put AS, he put AA. and he tried to look at the bands that were created as a result of the amino acids that were identified in the primers that were used when he looked at glutamine and valine, which are the amino acids implicated in sickle cell disease. And if you look at that, when we say most people know what is AA, they know what is AS, they know what is SS. Just know on this platform, from this webinar today, that this application of high-level state-of-the-art study, mechanistic study using genomics, human genomics and animal genomics, is able to make us have what we call gene therapy for sickle cell disease. As we talk presently, there is no cure for sickle cell disease. Sickle cell is a genetic disorder. Therefore, some scientists are of the school of thought that the only treatment for sickle cell will be genetic in nature. I don't belong to that school of thought. I belong to the school of thought that the treatment for sickle cell is natural, because it is a natural displacement of one amino acid by the other. Therefore, there must be something in nature that will help alleviate this. And that is why, where, for example, I work, we have a product for sickle cell disease, and it is the first herbal medicine that was ever registered by the United States of America, FDA, as an orphan drug for the management of sickle cell. We call it Niprisan. And those of you who don't know may want to Google it at the end of this meeting. Today, a big pharma company in Nigeria is marketing that product. But from what Ferez presented, you notice that he gave us statistics, for example, of how sickle cell anemia is just about 100,000 people in America, while 92% of sickle cell cases are in Africa. So you can see that it is an African problem. In fact, we call it a black man problem, because even the 100,000 that we are talking about, if you go to US, that 100,000 is mostly in the black population. So that's why we call it a black man issue. Now, the molecular basis for sickle cell, and please, can the host stop muting me? I just got a message that I was muted again. Professor Musa?
I didn't know you were Simisola, sorry.
I'm the one speaking. Okay, thank you. All right, so 92% of sickle cell cases are in Africa. Therefore, it is an African problem. But Professor Musa, can you see the link? And I'm happy that you are part of Ferez team. Can you see why science, when we talk to our mentees, we tell them that science is about networking. I'm glad that you are part of Ferez team, but it is an opportunity for you and IBB University and all of us to network, to collaborate, to partner on bringing home the application of genomics to the treatment and providing solution to this problem that we can see is clearly an African problem. So we now have the molecular basis, we know that, but there are some innovations that Ferez has brought out this afternoon, and I'll quickly talk about them, Now, but before I do that, he mentioned something about sex determination. And if you look at his slide in the YSR expression, you notice, I'm sure for those who understand it, it was interesting. For those who don't understand, let me say it in a local way so that you understand that one of the useful applications of genomics is in sex determination. You see, in those days, these things were considered aberrations or abominations. They are no longer, if you want a male child, genomics, you can do that. If you want a female child, genomically, these are all possibilities now. Bearing epigenetic factors that may be responsible for one thing or the other, but scientifically, all of these are possible. Now, but there's something that I want us to also, especially scientists on this platform, to take note. I want you to take note of what Ferez said, especially when he was talking about the SRY locus in the somatic and germline tissues variabilities. Notice the variabilities and the fact that he said that some of those things they are still unable to explain. I want everyone, all scientists, to note that inability to explain something scientifically does not mean that that thing does not exist. And this is where some of us, African scientists, are very vocal. Because there are a lot of things that you cannot explain. Because you cannot explain does not mean that it does not exist. It does exist. Your knowledge is limited. You are unable to explain. So continue to struggle to get the explanation. While you are struggling to get the explanation, the biological effect which you see, which we try to encapsulate under what we call systems biology, you continue to pursue it and allow it to go. And that is the principle that is used in countries like Africa, China, and India in traditional medicine. For example, I come from a traditional village called Agaliga in Olamaboro local government in Nigeria, where if someone has a compound fracture or a fracture that needed amputation, the local people in the community can still take care of that fracture. And sometimes what they do is they just break the leg of a chicken, and as they are manipulating the leg of the chicken, the human being is crying. And then someone is there because he's unable to understand it, is criticizing it. Your knowledge is limited. You cannot understand it. Okay? So calm down and try to understand. And so I'm glad that today Ferez from the US has told us that there are some things that even in these wonderful studies that I've done up and now there are things that I couldn't explain. Some of the innovations that I would like to mention, the pull down assay is interesting to me and attractive. And I think Professor Musa will, at the end of this meeting, begin to put up mechanisms by which we can all engage Ferez in this area and how we can use this to provide solution to problem. Because if you look at the comparing, Western blot, analysis with the quantitation that came up from that pull down assay, even though they are late, with the high specificity and selectivity, especially when he was talking about the DNA recombination, model, it is something that I think we can probably begin to see how we can utilize this, apply this in several of our own studies here, especially in natural products discovery and development, to see how, and I'm sure Ferez should be interested in this too, because this would be like extending the scope of what he has done in order to see how validation can come up and how it can be more applicable globally. The EMSA, the electrophoretic mobility shift analysis, is interesting too. I will encourage people to read more about that. I'm not going to drop this mic without talking about challenges. But before I talk about that challenges, do you guys, can you still remember he talked about the process, the developmental process, kind of from evolution to omics to artificial intelligence to optics, specifically the use of biosensors? If you notice that, there's something I want everybody to take home. The fact that the use of mice, the use of rodent was consistent in his studies. So even with all the modelings, even with all the computer activities, even with all the genomics looking at in vitro studies and predictions, the use of rodent was key, and for me, very important, That's why today we cannot take any drug product to any humans without providing preclinical data. Preclinical data in animals, the studies that you did in animals, it is fundamental, because nobody is going to allow you to test anything in a human being, because human beings are human beings, animals are animals. If your new product, if your invention, if your discovery, if this investigational new drug or new product, if we don't know the safety, if we want to know how efficacious it is, if we want to know how good it is, we cannot just jump to human being. We will first want to know what happens when that is done in animals. So take note of that as we all run towards the technologies of bioinformatics, artificial intelligence, machine learning, and all of that, which appears to be turning people's head today, as no matter what we do, we cannot remove the fact that studies in animals will be key to whatever we do. So the challenges that he mentioned, I'm happy that he mentioned those challenges. So his lecture, his 40 to 45 minutes lecture was all encompassing. And I'm glad he was able to touch all the areas bearing the itches. He talked about lack of communication between scientists and the background itself. And this is what we call the missing linkage between the academia and the industry. The industry here represents those that will apply whatever we do in the lab. And you see, it is that application that is useful. Whatever we do, whatever discovery we do, no matter how wonderful our papers are, our publications are, our patents are, no matter how wonderful, if it cannot be converted to the form that our people can use, then we are truly failures. If we are speaking on Zoom this afternoon because that technology has been converted into the form that we can use practically. That's the reason. We can hear Ferez from US because it has been converted into the form that can be used. Ferez can fly to Nigeria through the aeroplane because after the studies that discovered the radar in King's College, it was converted into the form that it can actually be used to direct and to monitor the aeroplane to fly from one place to another without crashing. So whatever we do, no matter how wonderful, no matter how wow our discoveries or inventions are, without electricity, I don't think we'll be able to carry out this webinar. So if the discovery by Faraday and his colleagues in those days didn't culminate into this tangible thing that we could see, then we will all be in our respective villages. Therefore, it is a call to action for all of us, And Professor Musa, I'm challenging you to lead in this because you have started very well. And I want to especially commend you for this and your university. You will need to, because there will be many challenges as you move ahead. So lack of exposure is also one of the challenges. And that is one of the things that this webinar has brought. Let me tell us, for those of us who may not appreciate this, if you had to travel to the United States of America to listen to this man that has spoken this afternoon, I believe many of us on this platform wouldn't be able to go. And that is what Professor Musa and IBB have brought to us this afternoon. And we should appreciate that and ensure that we do not fail ourselves. But this is one of the things that lack of exposure will help to fill in, the gap that it will fill in. Because if you stay in your country, let me tell you, whether you are an American or you are European or you are an African, if the only country you know in the world is your country where you were born, you are as good as the man that was born in my village and never traveled anywhere. So exposure is very, very important. And I believe this seminar series is one of the ways by which Dixon and his group will be bringing that to us. So communication is key. Linkage with entrepreneurs and industry is key. And I believe that if we do this, we will be heading with, finally, I like to talk about COVID-19 anytime I have opportunity. For the past 2 years, I've had opportunities to speak or chair more than 200 webinars. And I always make this comment. When COVID-19 started in November 2019, by December 2019 and January 2020, there was prediction but that 50 million Africans were going to die and that there will be corpses on the street of Africa. The prediction did not come to pass because Africans have been protected by what is available in their environment. The challenge, therefore, is before African scientists and African political leaders to ensure that whatever is hiding in our environment that must have been responsible for this protection is harnessed to ensure that we have products developed towards future COVID, whatever, because there will be many epidemics or pandemics in future. Having said that, I would like again to thank Ferez very well. We are grateful and want to thank you for waking up early to make this presentation. It was an eye-opener. It was an exposition for us. We have all learned, and I hope that when we call on you again, you will not hesitate to be available to us. Thanking also Professor Musa, because I believe this is my last opportunity to speak. Thanking also Professor Musa and his colleagues at Ibrahim Badamasi Babangida University for leading this and for ensuring that this came to us on a platter of gold. And for all of you who took time out of your Saturday instead of going to parties and Owambes, as we call it in Nigeria, you are here to rub minds with us. You will not be disappointed. Let us all join hands together to develop wherever we are, wherever we have the influence, for the future is in our hands. Thank you very much. I yield the podium back to Professor Musa, who is moderating. Thank you.
Thank you very much, sir. Yes, before, I think we can now go to questions and observations from our participants. But before we do that, I think Ferez has something to say. So I'll yield the mic back to Ferez to say something before we go to questions and answers. Thank you. Ferez.
Thank you very much, Dixon. Professor Image, thank you for your kind words and for making a nice summary. And yeah, I look forward to working with the Trans-Saharan Disease Research Center. But before I say anything else, I also should thank all of you, as Professor Image said, for making time out on a Saturday evening. I realize it's evening there instead of socializing, which is a great honor for me. And so I'm obligated to apologize because I now know that the most important and relevant part of my talk was on the other PowerPoint, but I'm going to make amends. Dixon, what we will do is, since it's still fresh in my head, is present it to the LSI and you record it, and then you can share it along with the new slides. Because in those slides, there was more about African science and why biodiversity, whether it's African. I see Professor Kolis is from Indonesia, who's part of our institute, also does this in Indonesian biodiversity. There are examples for the last 150 years where people have shown that there's large genetic and epigenetic changes that could be harnessed into a drug. The classic example is the heterozygote in the sickle cell anemia case, which lowers susceptibility to the disease by about 90% compared to the homozygote, and increases lifespan in, I believe, in Ghana, but I'm not sure, by 60%. So there's, beyond that, there are many, many things that's on the new talk. I don't know how this thing got onto, when I was transferring the slides, that's what happened. But we will make it complete and we'll send it to you. And thank you very much for being here. And I'm happy, Dixon, thank you for organizing this. It's been a great pleasure.
Okay, thank you, Ferez. I think some questions are beginning to come in, but if
Yes,
I may respond to what our chairman said before we go on, talking about COVID-19 and the fact that Africa was able to beat COVID-19 is despite the predictions to the contrary. At Trans-Saharan Disease Research Center, we also had such thoughts, and we didn't stop at thinking. We started to research into why Africans may have beaten it. And one of the things we looked at is our diet. And we decided to do a survey on our diets against the virus. And we discovered that there were 2 vegetables that we take regularly here that were zapping the virus very, very well. And in fact, in one instance, we had like 8 log reduction in the virus population after applying this vegetable extract. So what is stopping us from going further now is funding. So this is an opportunity to ask for collaboration and funding. We already have a protocol that is working, and we believe that if we have somebody to fund us, we can actually bring this to the market. And it may not only be for COVID, maybe for the next emerging disease, which may not take up to 5 years again before it resurfaces. Thank you very much. Now, yeah.
That's really nice to know. That's an important discovery.
Thank you.
And yeah, it's amazing if you get funding to do this and have collaborations somewhere. Maybe what you do in the field and in Nigeria is one thing, and what they do is another thing. And working together, you can come up with something much stronger. In other words, you already have the observation.
Yeah.
And you already have what you want to know is a mechanism. Like when Alexander Fleming discovered penicillin by accident, it's called serendipity, which is a big part of what our proposal is. You have natural progression of plants, and then you have accidents in the and collaborations and civilization that make it much bigger, right? There are also downsides. You have the Industrial Revolution, and now we have climate change. So you have to meet them, meet those challenges and the products. You are very well placed. I didn't know this work. My personal perspective is from a genetic end. In other words, they are, genetics and epigenetics. In other words, in the original slide, if we'll talk about that later. Let's continue this thing.
Thank you, Ferez and Professor Musa. What you have said, I'm glad that there's also something like that from your center and from your team. There are a lot of those kind of results currently in Africa. Professor Katere from South Africa is leading a huge group in Farmer Connect on that. And then in Nigeria, TED Fund has given a lot of funds. About 3 or 4 weeks ago, specifically in February, the Nigerian Academy of Science gathered about 6 awardees to make presentation on on this kind of natural products intervention in COVID-19. What is lacking, and which I want you to do, is sometimes those of us in this part of the world, we find it easier to collaborate with people in US, in Europe, than the people around you. Because if you look at our own environment, even in Nigeria, there are a lot of labs that do a lot of things, and sometimes we repeat ourselves. Actually, if we put our energy together, we will do more and we will do better things than everybody doing the same thing in different form or the other. So I will encourage you to and I'm going to be part of this to link up with institutions. For example, I mean, my institution is leading the mega TED Fund research of the Forestry Research Institute, the Pharmacists Council of Nigeria, for example, in drug development, which we, so let's link up with, Professor Happy of Redeemers University. He has done a lot in genomically. It's an African Center of Excellence for Genomic of Infectious Diseases. So if we all link up, all link up and we stop all this silo method of doing our own things, we will achieve a lot before we even talk of funding.
Let me take this opportunity. I see that Professor Collis of Indonesia, who's part of our institute. It's not my team, by the way, Professor. We are all partners in this. I couldn't do anything without everybody, and everybody has their own thing, so we are partners. I see Professor Collis Hauda from Indonesia on the talk right now. He's doing the same thing in biodiversity and making the same contributions.
Okay, great.
And so he should, you should perhaps reach. Dixon knows him. I think Dixon mentioned him. Dixon is my friend, so Professor Musa knows him, and they interact well. And this is a bit removed from my expertise, but they're in the field of COVID. In fact, the first paper that our institute put out was to basically say that we have 2 members of our institute that are doing work, that are at the front lines in Nigeria and in Indonesia that are working on COVID. So the major
challenge, the major challenge in COVID studies in Nigeria is not the early detection or prediction of the possible efficacy of our natural products, but the application, trying to get to the real effect, and where you need P3, P4 laboratories in order to work on the SARS-CoV-2 itself, because we don't have a functional P4 laboratory, even P3 laboratory. One, I understand one has just come up at the Veterinary Research Institute now, and everybody cannot line up there. So if you look at what Dixon and his group have identified, a lot of recipes, extract, what we call active crude extracts, or even purified fractions are available. So one of the things that networking can do is that in US, in India, in Europe, where there are available P3, P4 laboratories, we can easily partner together, and it's a win-win situation because we will be able to come up with interventions. Thank you.
Thank you very much. Let me go straight to the chat box now. The first question is from Aremu Haruna. He said, "Thank you, Dr. Ferez, for the wonderful presentation. I would like to further ask, can you highlight some established epigenetic processes that have been associated with sickle cell anemia?"
That's a very good question.
And also kindly give comments about proposed hypotheses to navigate some of these problems.
I'm sorry, what was the last question?
Can you highlight some established epigenetic processes that have been associated with sickle cell anemia? That is one. And he also wants you to give comments about proposed hypotheses to navigate some of these problems. Thank you.
Right. So I'll take the first question first. One of the best examples of epigenetics and genetics, I guess, is the locus control region of the beta globin locus, which causes sickle cell. In the beta globin locus, it's one code letter word change. The adenine to thymine, adenine to thymidine change at position 6 that codes for the glutamate to valine that causes sickle cell anemia. It's clear, like unlike say cancer, you can have a mutation and maybe it'll require cell burden, etc., etc. In this case, there's a clear link, right? The locus control region does not code for any protein. It's like an enhancer or a promoter. And so what is epigenetics? And epigenetics started with Jean-Baptiste, what's his last name? Lamarck. Remember way back, a French scientist. And it's gone through many, many steps. But the proteins that bind to the locus control regions determine the changes in gene expression, the switching of the embryonic, fetal, and alpha globin. I mean, embryonic, fetal, and adult globin, beta globins, right? They determine the level of the protein, right? So in the first constructs, they actually left out the locus control regions, partly because, one, it was difficult to put it into the viral vectors, and two, at that time it was too far away from the beta globin gene to really be like a conventional gene. It was 60 kbs away. So it was in that time called junk DNA. Well, now we know that where that junk exists, it ain't in the genome from many things like microRNA, et cetera, coming up. There are many other examples. What we showed are not conventional genetic epigenetics for the Y chromosome. They are more a global chromosomal effect where the covalent linkage, and I haven't shown the data because I took them out because you don't have time, but those mobility shifts that you saw are caused by structural changes due to the titrations with pH and also caused by the proteins that are linked there. So on the Y chromosome and all other chromosomes, there are proteins that are linked. And in the SRY region, there's a single amino acid change that can reprogram that can prevent that SRY gene. So instead of getting an XY male, you get an XY female. And one thing, important thing that I didn't say in the talk, in becoming flustered, why can we see all these changes in the, why can we see these? I'm going to use a colloquial term because Professor Image reminded me that not everybody is in this field. The gene breaks. Well, if the gene for sex determination breaks or disappears, how do you get an XY male? Well, the real answer came from my academic grandmother, Professor Beatrice Mintz, who used to work with allophenic mice. Basically, you take two mice that are genetically defined embryos, stick them together. As the mouse grows, you can see the cells from mouse A talk to the cells from mouse B. It's going to be a brown mouse or black mouse, etc. You see the colors in the coat, and you can see this in the coat colors. So that Y chromosome is only needed from day 10 to day 11 and a half. This was done by Professor Robin Lovell Badge, but it was done even before him by Dr. Beatrice Mintz. And she showed that you could have this allophenic mice or chimeric mice in which 95% of the cells, somatic cells, are XX, female. Only 5% are male, XY. And yet that animal became a male, a sterile male, because you need genes for spermatogenesis. So what that means is that the gene for sex determination is not needed in adults, which is why we can look for it. This has also been done by Dr. Lalji Singh and Kenneth Jones at Edinburgh, where they worked with a mouse in which 90% of testicular cells were XX and only 10% were XY. So male, female, as Professor Image said, is is an equilibrium, and we all have one or the other in us to some extent, right? To come and tell you, so that specific gene is that SRY gene. But there are many other cases. President Shelley Tillman of Princeton University has worked at I believe the interferon locus and shown imprinting. If it comes from the paternal or the maternal germline, it's differently regulated. There are also, I'm trying to say, Davor Scholter showed androgenetic and gynogenetic embryos. Basically, if you took out the nucleus from either the father, or you took out the nucleus from either the mother, and you know, these mouse biologists are great at doing this, and put in another nucleus that was so two mother nucleus or two father nucleus, that embryo gets all kinds of developmental diseases, bone problems, orthopedic, regulatory problems, and many of them are lethal. So you need both dad and mom. Now they made a mouse with only the dad's genome and mom's, or only the mom's genome subsequent to that. But I don't know how they did it. I didn't follow it. For us, sex determination is mainly a target, like it's been for scientists for 100 years. And that too, I left out in my talk in a hurry, right? So all of these are epigenetic and genetic. And final thing that we need to say, it's clear from cancer. You know, many people switch to epigenetics or genetics. Actually, one of my mentors, Professor Alexander Rich of MIT, I never worked in his lab, but I consider him a mentor. He was the one who finally put it in my head that the structure of DNA can have a regulatory effect. In other words, it's not just floating there. It's not just read out like a zipper. It forms 3D structures. That we're learning more about it today. He was the one who came up with left-handed DNA. Well, what he liked to say was that there's a fashion in science, right? So many of these things that you raised, right, are now becoming recognized and becoming established in the field. I don't know if I answered your question, but I certainly tried.
Aremu Kolawole, can you come in? Aremu Kolawole. Okay, we go to the next question.
Okay.
And the next question, I think I will want more than one person to answer this question. I would like colleagues to speak on this question too. I would like Percy to speak on this question. Professor Emeje too. I think you can come in, but I think I'll let Ferez go first. And this is the question.
So I can make my comment very brief. It's totally out of my experience, totally out of my expertise.
Okay.
So, you know, if I say anything, it'll be wrong.
Okay. African traditional medicine in Africa denied chances of acceptance in the global north because they are from a land of lower technology, because they don't actually cure disease, because African medicine on the market will impact pharmaceutical companies in the global north, or other reasons. So this person is this Kelly Allen. Thank you, Kelly, for joining this meeting. You joined very, very early. And the question is, why are African medicine denied access in the global north? Is it because our technology in Africa is poor, or that the medicines from Africa do not work, or that it will outcompete the pharmaceutical companies in the global north, and so they must not be given a chance? So what can you give a reason for why you think African medicines are being denied access in the global north? I think Ferez, you can go first.
You put me in the hot seat, sir. There are two parts to the question. Everything that you raised is almost certainly a contributing factor. I know this from my own experience, and I'll tell you, I'll move it out of the African context per se, so you can see it on a more stratospheric version. The traditional medicines, whether they're African or Mexican or Indian, are probably not given the same opportunity. Now, I know one drug from India, which was part of thousands of years of history, Rauwolfia, which is good from a herbal method called Rauwolfia, was tested and found to actually do what the Indian traditional medicine said it did, which was to delay many things, heart attacks and stuff like that. But they looked at, FDA looked at Chinese, many Chinese extracts and Indian extracts, and they couldn't nail it down. Now, that might be a technical problem because some of the people who worked on herbal extracts mention that there's a lot of variation from extract to extract. How you extract it makes it different. So the chemical composition, how you test it makes it different. And I believe all of that. So it's partly everything that you say, partly not. The second part to that question is, and I've had a good dose of it over the last 40 years, shows how stupid I am, but to stick around instead of doing what my brother is, go make some money. But the whole issue of reproducibility, right, can be a double-edged sword. So there's a paper out there saying that 70% of papers in science are not reproducible. Now, my immediate reaction, if it was strictly personal, that means from my own experience, which can never be all of history, all of civilization, thousands of years of civilization, it can never be representative of that, would be to agree. And it's not just in ordinary journals, but in top journals like Cell Science, Nature, PNAS. But let's look at that. If 70% of science were irreproducible, and science has been driving civilization for thousands of years, would you have such things as the Renaissance, African and European and every others, the Industrial Revolution, the Information Age, the Space Age, the Green Revolution, the Biotech Revolution? Think of all the mom and pop things that go on around the world, all the Apples and the Mac and the PCs and, you know, what's it called? What's Bill Gates' company called? You know what I'm talking about. All these Microsoft, US Steel, all these things happen. That's a stratospheric view of it, right? So I'm certain that African science is being left out from my gut feeling. Second part is, what do you call reproducible? What do you count as reproducible? Any bench scientist knows that you use thousands of things that are manufactured in factories, from pipettes and weighing boards to, I mean, solutions that you make on the bench, the cells that you grow, media that, you know, it's way beyond limiting. Do you count those in reproducibility or do you exclude those, right? And why? And the third thing is your technical expertise. There are many scientists, you know, I want to tell you about that assay, you know, that assay I showed you on the pull down assay. Yeah, it disappears for no reason, but it works with controls and it gives you that same result with that assay or another assay. And yet somebody who is very influential in that said it's not reproducible. That's not true. It is reproducible. It doesn't give you the result you like, but it is reproducible. I'm sure African science has that same experience. So, you know, reproducibility has a double-edged sword. And sometimes there was I don't want to say this name because there's some other work that came out that actually confirmed what was said, but there are many who can make things work. I know that I can't make work. If you ask me to do, you know, abstract mathematics, I'd flunk. Einstein didn't do very well with mathematics. Bose-Einstein equations came up, right? The Indian scientist Bose, Satyananda Bose had to be on his equations to help him solve those. So a lot of it is, you know, what you said, the silo view in science actually hurts science. It helps science. I think the Nobel Prizes are great. I believe in CRISPR-Cas9 and CART and all these amazing things for which the prizes have been given, but they will never move the world where it needs to go, which is to stop those diseases from starting in the first place. So which is better? Trying to cure cancer or never letting it start? It's easier said than done, but that's where I leave it. So I didn't answer your question because I can't, but I think that you will recognize from my answer that we have had similar perspectives in some ways.
Thank you. Colleagues, can you come in, please? Professor Evans, I will need your perspective on this too. Professor Collis, can you come in, please?
Yes, speaking.
Yeah. What's your perspective on this? That the question is, what do you think is responsible for medicines from Africa not being accepted in the global north? What is your own experience from Southeast Asia?
All right, thank you. First and foremost, I would like to thank Dr. Musa for inviting me to join this wonderful meeting, and always to Dr. Ferez. Prof. Ferez has always have a wonderful talk. Thank you for a very good insight. So I think the issue is quite similar between Africa and Asia in general, Dr. Musa and all fellow colleagues, that the medicines from, well, there is always a controversy between modern medicine and traditional medicine, right? I think. But if we look at a bigger picture in this battle between modern science or modern medicine and traditional medicines, because actually in the reality it's not that so, because if we notice, for example, it really depends on what stage we want to see. Yeah? For example, NCI actually what they do is collect 230,000 extracts from around the world. So by having the extract is the first step toward the discovery of drugs. And as you know, that this NCI, the 230,000 extracts, they collect from all around the world. And most probably, some are from Africa, some are from Asia, from Latin America, and so on and so forth. Because, sorry to say that European or American actually not so much rich in terms of biodiversity. But those countries in the sub-equator and subtropical, they are much more richer, yeah, in terms of biodiversity. So, but this fact that they collect pretty much everything from around the world and end up with a collection, a unique extract collection from all over the world, is the fact that actually the modern science, modern medicines, if we can maybe refer to this as American or Westerners civilization or society, actually they heavily rely on the resources, these resources, yeah, which is based on, for example, local wisdom, traditional medicine that have been practiced for so many generations, yeah, all over the world, yeah. Not only in Africa, but I would like to say also, including Indonesia, for example, as one of the mega biodiverse country, we have so much to offer in terms of traditional medicine or natural products, yeah, in general. I myself established what's so-called Indonesian Natural Products Library, yeah, which is actually the first of kind in Indonesia. We try to adopt actually what had been done in NCI, and also with the European Bioinformatics Institute. Yeah, what I'm sure many of you are familiar with Campbell, yeah, chemistry, and European Molecular Biology Laboratory, but for chemistry, they do very similar works as I do. But of course, I just started several years ago. Yeah, nothing compared to Campbell in terms of the collections. But what I'm trying to share with you here is that we have the potential. It's just the way how we later on follow up. Yeah, we cannot just stop until the extracts. Of course, as I mentioned earlier, that extract is just the first step in drug discovery in general, because from extracts, from fraction, for example, we can pretty much have a very good start in terms of drug discovery. But of course, the science, we understand that the science, the leading countries still in America or in European in general have the advantage of this science and technology. They have all this technology or the expertise that is required in terms of following up the discovery of the drug from natural products. So this is, I think, the things that we need to share. The developed countries need to share with us in the third world countries or developing countries. Yeah, I'm sorry if I said third world countries, developing countries, that we have the resources. Yeah, please don't try to deceive the world. That you guys in the Western, they don't need us. But you look for us every single day in different part of the world. That is the fact. The NCI has a collection of 230,000 unique extracts. I can share you the paper. Yeah, this the American Chemical Society of Biology, the paper. So, meaning that in the reality, in the practicality, in everyday life, that they do look at this traditional medicines. Otherwise, when and where will they start from, if not from this point of view? So that's why I think Africa, which a very rich culture, tribes, as well as Indonesia, for example, have a very good advantage, in terms of promoting our own traditional medicines. But of course, we cannot just stop until there, but we have to follow up pursuing a more scientific or technology proven research. Yeah, we have to follow this up. But we have a good starting point actually compared to other parts of the world. I think that's maybe I can just give a very general comment, Dr. Musa, and of course it will can give you get something out of it. Thank you.
Thank you very much. Thank you very much. Professor Evans, can you come in, please?
All right, thank you. Can you hear me?
Yes, we can.
Yes.
All right. So it's a good thing to discuss this very topic. It's a topic that is heavily debated all over. Particularly those of us who are in Africa, we are passionate about this topic. Well, the basic thing I will want to talk about, or I think is from my own opinion, is the economics, pharmaceutical economics. That's my own area of take. I think that the global north is looking at the economics of developing African medicine. Now, my experience is that if you have any extract or any molecule that's very active, it will not go far until you involve a researcher from the global north. It will not go far in acceptance. Agreed that our technology may be a little lower, but accepting that it is effective is one thing. And then developing it from the low grade to the next grade is another thing. So my take is we must find a way to bridge that economic politics involved in pharmaceuticals, thinking of who is recognized, who makes the money, who is the one that takes the credit of this invention. I think that's a real thing. Thank you.
Thank you very much. Percy, can you come on, please? Percy, do you have a perspective to this? Is Percy on?
Yeah, his mic is muted.
Okay.
Yeah.
Professor MJ. Mr. Chairman.
Thank you, Professor Musa.
Quickly.
Thank you. I will try to be short, and I believe people who read about me in the news know my perspective as it relates this. And when I talk about this area, I also talk authoritatively because it's my area of expertise. Not just because I'm a pharmacist, but also because I've had the privilege of being the leader of all pharmacists who teach in Nigeria universities and research institutes and try to bridge the gap between the academia and the industry in order to see product develop and go to the shelf. But this is what I have to say on this. First, it is important for all of us Africans to stop blaming the West. Stop immediately from blaming the West, because the acceptance is not even there at home. So why are you blaming the West? So have you accepted it? Most of us who are scientists of high repute today grew up in villages where traditional medicines or our plant biodiversities were used to cure our diseases. But today, we have all traveled to different parts of the world. So we now know that there's one country called developed country and another one called underdeveloped, or first world, second world, third world, and all those nomenclatures. So I call it 21st century slavery mentality. When we have to decide, because you see, the truth is, if you go to China and India, and I'm glad that Ferez made reference to Rawofia, even Taxol that India lay hand to today, which is a powerful anti-cancer agent, is from natural product. In fact, I can tell you as a pharmacist that at least 40% of the drugs, of the conventional drugs, the orthodox drugs behind the pharmacist in the pharmacy in the Western world are from plants. The National Cancer Institute in US has said that about 70% of drugs today in the last 25 years were developed from plants. WHO has said that about 80% of drugs that are used today are of natural origin. Therefore, our problem is not that the West is not accepting it. No, we need to deliberately devise mechanism to make the West accept it. And that is what China is doing. That's what India is doing, whether the West likes it or not. If you go to Tianjin University of Traditional Chinese Medicine in China, you will see a lot of Americans and Europeans studying medicine, acupuncture, and TCM. If you go to India, you will see, if you go to Pune, Maharashtra, you will see a lot of Europeans. In fact, we call Pune the Oxford of the East. You will see a lot of Europeans and Americans studying natural products research there. So when we decide to begin to value ourselves, to have confidence in ourselves, the science is science. There's no low technology and high technology. Technology is technology, and it's available to all of us. The technology that I use here in Abuja, Nigeria, is what is used in US. They are not different. In fact, this year, the two people that were taken by Erasmus Mundus in UK to come for fellowship are from my lab in Nigeria, the two Nigerians that were taken. So we as Africans, we first of all have to develop that confidence. We need to do standardization. Whether they are extracts or not is not the issue. You can call it any name you like, but if you do standardization, dosage, preparation, and delivery, if you have monitoring parameters to monitor what you are talking about, if you develop the system biology approach to monitoring the efficacy of what you are using, then you have what we call the drug dossier. The dossier, that is what you will use in phytovigilance. That's what we do. That's what we need to do. But as it is right now, even here in our own country, among the healthcare professionals, we are all confused because, no, we cannot use this, you cannot use that, you don't know the mechanism of action. I'm a pharmacist. What is the mechanism of action of propranolol? I still teach conventional drugs in the university, and we still tell our students that some of these chemical drugs, the mechanism of action has not been fully elucidated. So what is the problem if you don't know the mechanism of action of how fractures are treated in using herbal medicine? So, but we need to do proper documentation, standardization, develop our dossier in a way that when we say that this particular plant, this is what it does, this is the dose that we do this is the side effect. And once there is an overdose, once you take more than necessary, this is another plant that you can use to call antidote to take care of that. Once we have all of that properly documented and form into our own pharmacopoeia, the way there is Chinese pharmacopoeia, the way there's Indian pharmacopoeia, then we will be good. Then the people we are complaining of, Europe or America, they will now begin to look for our products. They will begin to look. Nobody is going to tell you to bring what he cannot understand. That is my contribution.
Thank you very much. Dr. Ilo, you have something to say, please? Let's do that quickly.
Yeah, thank you very much, Mr. Musa, for having me. Thank you so much for all the presentations and all the contributions. Mine will be slightly different from what everybody has said. I think the West needs African medicine, as a whole. I remember my days working in Professor Iwu's lab. All our herbal products, we had American pharmaceutical companies coming to pick them. But the only unfortunate thing we noticed, and it's still happening, is that once those extras are structurally elucidated, they abandon the natural product scientists because they only have the structure. So they just start synthesizing it because they have the structure. So that is the problem, and that is still the problem till tomorrow. So if it's certain new compounds, new products, the Africa has it because we have the plant biodiversity. Most of our biodiversity has not been known. Most of our plant uses have not been known. So there's still a lot out there. And so these pharmaceutical companies, the Western world, will still look up to Africa for herbal products. But the thing I would like almost of natural products scientists to tackle is the fact that when it gets to an elucidated structure, the pharmaceutical companies abandon those natural products scientists because they already have the structures. Thank you.
Yeah, you patent it. You patent it and you hold the pharmaceutical companies to ransom. They have to contact you before they do anything. It's been a very, very, very wonderful session. I think most of the questions in the chat box have been taken. And we were supposed to round up by 2, but this is 2:20 p.m. Nigerian time. I want to especially thank Dr. Nallaseth for waking up by 6 a.m. US time, even though by 1 a.m. we're still chatting. He had not gone to bed. He was still able to wake up to give this presentation. And I know he has been busy, and also he was under the weather for some time. So actually giving this talk today is something wonderful. We are very, very grateful. He has tried to compress 48 years of work into a 40 minutes presentation. That is a very difficult thing to achieve, but thank you for trying. We also want to thank the chairman for this webinar series, for this particular webinar, Professor Emeje, who brought his experience and expertise to bear in handling most of the talk today. Thank you very, very much. He was the first to join the session today, then followed by Percy. I was hoping Percy could talk, but it's as if he doesn't want to talk today. Kelly joined us, and eventually everybody came. A lot of people contacted me that they could not join because of some technical issues or the other. We'll try to correct this before the next webinar series. But you all agree with me that this has been an excellent and exciting session we've had today. Thank you very, very much. We'll bring this seminar series to an end, except if somebody has something quickly to say. If there's any such question, observation, please, let's have it quickly. Okay, in the absence of none, let me thank each and every one of you again for honoring our invitation and joining up and spending about 2 hours, 30 minutes, or in some cases, 3 hours, spending your data. Data is very expensive in this part of the world. To join up, it's a very, very wonderful thing, and we appreciate it. The next in this series will come up soon. We already have your email address. We will contact you before the next series come up. Thank you very much. Thank you, Mr. Chairman. Thank you, Mr.
Thank
Speaker.
you.
God bless you.
Thank you, and congratulations to you and your team.
Thank you.
Thank you, Prof.
Have a nice day.
Bye, everyone.
Bye-bye.
Thank you so much.
everybody. Yeah.
Thank you, everyone. Thank you,
Thank
Dr.
you.
Musa, Dr.
Thank
Thank you,
Ferez.
Dr. Musa
Thank you
and
very much.
so
Yeah.
much.
Professor Image. Thank you very much.
Thank you, Ferez.
Have a great day, everybody, and thank you for being
Thank
here.
you, everyone. Thank you for coming around. Thank you,
Bye,
Professor
bye.
Ferez.
Ferez, have a nice rest.
You too, Professor Carlos.
Okay,
Yeah.
take care. See you. Bye.
Professor Agu, thank you for joining us. Thank you very much. Okay, bye, everybody.
Bye-bye.
Thank you, Professor Mut, for what you are doing. Congratulations.
Thank you, thank you, thank you.
Bye-bye.